Welcome to Life Science Library. In this video, we are going to see about another secondary metabolite, that is phenolics. So far, we have seen about terpenes and basic information of secondary metabolites. Now, we are going to see about phenols. In phenols, we are going to see about the basic information about phenols, various classification, various classes of phenols. Specifically, we are going to see about phenyl propanoid, coumarin, lignin, and flavonoid and tannin. This topic becomes very important for CSIR net preparation. There are so many previous year questions that came from this. Entirely secondary metabolite is more or less like a memory based pointer, and anybody from any background can read this topic and can easily score marks when it comes to CSIR net life science examination. Memory based questions can come from this topic, from the entire topic of secondary metabolites. First up, we are going to see about what are phenols. What are the basic information about phenols? Phenols are chemical compounds that are characterized by aromatic ring. They have one or more hydroxyl groups. So we know the structure of phenols. We have a ring, benzene ring, OH. This is the basic structure of phenol. And it is the most abundant secondary metabolite. It is the most abundant secondary metabolite in plants. This is a previous year question. Which is the most abundant secondary metabolite? It is phenols. They exhibit absorption at electromagnetic spectrum and visible region. This is also a previous year question. At what spectrum that phenols exhibit absorption? In electromagnetic spectrum and visible region. It acts as a defense against UV radiation, against herbivores, against pathogens. They give mechanical support. They help to attract pollinators, reducing the growth of competing plants. They help in fruit dispersal. This is the structure and uses of phenols, phenolic compounds. Next, we are going to see about various classes of phenolic compounds in plants. First is simple phenol. Simple phenol, 6-carbon compound. Phenolic acid, 7-carbon compound. Acetophenone, 8-carbon compound. Phenyl propanoid, hydroxycinnamic acid, and coumarin. C9, 9-carbon compound. Naphthoquinone, 10-carbon compound. Xanthone, 13-carbon compound. Anthraquinone, 14-carbon compound. Flavonoid, 15-carbon compound. Lignin, 18-carbon compound. Biflavonoid, 13-carbon compound. It is 30-carbon compound. Please make a note of it. Simple phenol 6-carbon, phenolic acid 7-carbon, acetophenone 8-carbon, coumarin, this is very important, 9-carbon compound, naphthoquinone 10-carbon, xanthone 13, anthraquinone 14, flavonoid and lignin, very important, flavonoid 15, lignin 18. First, we are going to see about phenyl propanoid. Structure of phenyl propanoid. We have a ring structure. with that attached with 3 carbon side chain. This is phenyl propanoid. It is a simple phenolic compound with a benzene ring attached with 3 carbon side chain. Transenamic acid, coumaric acid, caffeic acid and fulic acid are the few examples of phenyl propanoids. So phenyl propanoids comes under C9 category, consists of 9 carbon atoms. Lignin 18 carbon atoms, flavonoid 15 carbon atoms, anthraquinone 14 carbon atom. Okay, so phenyl propanoid is 9 carbon atom. Biosynthesis of phenolic compounds. Phenolic compounds are generally synthesized from shikimic acid pathway. Shikimic acid pathway. Shikimic acid pathway converts simple carbohydrate precursor and phosphoenol pyruvate, that is erythrose 4-phosphate plus phosphoenol pyruvate in shikimic acid to phenylalanine. Phenylalanine by shikimic acid pathway. Phenylalanine is converted into transenamic acid. It involves a process of deamination. Phenyl ammonia lyase is the enzyme involved. Phenyl ammonia lyase is the enzyme involved in this conversion of phenylalanine to transenamic acid. Transenamic acid is then converted into simple phenolics, other phenolic compound. This is the basic biosynthesis pathway. So, erythrose 4-phosphate and phosphoenol pyruvate combine and undergo a shikimic acid pathway. With shikimic acid pathway, they form phenylalanine. Phenylalanine is then converted into transenamic acid and then to phenol. In this conversion, deamination takes place. Phenyl ammonia lyase is the enzyme involved. Shikimic acid pathway. Coumarin from cinnamic acid. Phenyl propanoid lactone, it is called. It is belong to a family of phenyl propanoid lactone. Phenyl propanoid is 9 carbon. So, phenyl propanoid lactone. This is the structure of coumarin, phenyl propanoid lactone. Scoptilin. Dicoumaril is an anticoagulant. Alpha toxin is a fungal toxin. What is scoptilin? Scoptilin is probably the most widespread coumarin in plants. It is produced from cinnamic acid. This is very important. Phenyl propanoid lactone. Next we are going to see about lignin. Lignin or heterogeneous phenylpropanol. It consists of three simple alcoholics, that is coniferyl alcohol, coumaryl alcohol and synaptyl alcohol. Three alcoholic compounds. These are called as monolignol. It is the second most abundant in plants after cellulose. Biosynthesis of lignol is not understood. It is believed that these alcohols are oxidized by peroxidase. These free radical are oxidized into free radical by peroxidase. Alcohol are oxidized into free radical by peroxidase. Peroxidase is the enzyme involved. Then in cell it is present in the cell wall. Secondary walls of trachea elements in xylem. It is xylem component. It is involved in protection in plants. Protection, deter from herbivore feeding. Blocks the growth of pathogen. It has various general functions. Protects from herbivore. Herbivore cannot digest it, so herbivore can never eat. Block pathogen growth. It provides rigidity and durability to plants, and it provides protection. So lignol, alcoholic compound, is converted into free radical by peroxidase. Peroxidase is the enzyme involved. Next about flavonoids. Flavonoids, we know they are 15 carbon compounds. Here we have C6, C6, C3. They are 15 carbon compound. This is the basic structure of flavonoid. Then substitutes such as hydroxyl group and sugars are very common. Flavonoid exists naturally as glycoside. These are anthocyanin, flavanone, flavanol, isoflavanone. They undergo they have a property to undergo. They are capable to undergo hydroxylation, methoxylation, prenylation or glycosylation. By all these processes, they form variations like flavanone, flavanol, isoflavanone and anthocyanin. They can undergo only these chemical reactions: hydroxylation, prenylation, glycosylation and methoxylation. Next we are going to see about tannin. Tannin are water-soluble plant polyphenol. They are hydrolyzable and they are condensable. Hydrolyzable and condensed. Two types. Hydrolyzable and condensable. Condensable form is non-hydrolyzable, hydrolyzable and condensable. In contrast to polyphenolic polymer lignin, tannin are located in vacuole. They are phenolic polymer living in the system. They are living components. They are polyphenolic polymer present inside the living system. Polyphenolic polymer. Hydrolyzable tannin. Gallic acid is an example. Hydrolyzable tannin include sugars like glucose with its hydroxyl group esterified, like sugars like glucose with its hydroxyl group esterified to form gallic acid like phenolic acid. It forms a cross-linked polymer. Condensable tannins are compounds formed by polymerization of flavonoid. These are also known as proanthocyanidin. Hydrolyzable tannin is made from esterification of glucose. This is the point. Condensable tannin are made from polymerization of flavonoid. This is the key point here. Okay, so far we have seen about complete details about phenolics that are most required for CCRNet Life Sciences exam. Now we will sum up the basic structure of phenol and its uses. We have seen most important is they exhibit UV and magnetic spectrum and visible region absorption, and they are most abundant. It's a previous year question. Next, simple phenolic 6 carbon, phenolic acid 7 carbon, acetophenone 8 carbon, phenyl propanoid 9 carbon, lignin 18 carbon, flavonoid 15 carbon, anthocquinone 14 carbon, xanthoquinone 13 carbon and naphthoquinone 10 carbon. This can come in pointer. Cinnamic acid C9 carbon, coumarin C9 carbon, phenyl propanoid, example transinamic acid, coumaric acid, caffeic acid, fulic acid, that is a ring with 3 carbon attached. Biosynthesis, we have erythrose 4 phosphate plus phosphoenol pyruvate. They undergo shikimic acid pathway to form phenylalanine. Phenylalanine undergo deamination to form transinamic acid. And the enzyme involved is ammonia, phenylalanine ammonia lyase, and they form many compounds like phenolic compounds. This is by shikimic acid pathway. This is a previous year question. Next we are going to see about coumarin. These three important. Alpha toxin very important. We will be seeing this in molecular biology as well. It is a fungal toxin. It is an anticoagulant. It is the most abundant coumarin. Most abundant in case of coumarin. Lignin is the second most abundant in plants after cellulose. These are the three simple monolignol. Alcohol alcohol is oxidized to free radical with the help of enzyme peroxidase and they form lignin. They help in secondary wall formation, cell wall formation, They are heterogeneous phenylpropane. Flavonoids are 15 carbon compounds, that is 3 carbon here, 6 carbon here and 6 carbon here. This is the basic structure of flavonoid. Tannin are water soluble plant polyphenol. Two types. Hydrolyzable and condensable. Hydrolyzable compounds are made from esterification of glucose to form gallic acid. Condensable form are formed by polymerization of flavonoid. In case of flavonoid, they undergo hydroxylation, methoxylation, prenylation and glycosylation to form many isomers of flavonoids like flavanone, isoflavanone, etc. and other compounds of flavonoids. So, we have seen in detail about everything about phenols that are most required for Gateiiunit life science examination. Hope this video is very helpful and very clear to all the candidates watching this video. And thank you for watching this video. Thank you.